Hello. Good afternoon. I think we should get started. My name is Jude Uzona. I'm the Vice Dean for Research at Radio Faculty of Health Sciences. And I will be moderating today just to remind you that the University of Manitoba campuses are located on understanding what they went through and also how I can be involved in trying to make changes in this reconciliation process. So I just want to welcome all of you today to the Gairdner lecture. I don't know how many of you have an idea about what this is all about. You can go and check. About a third of individuals who win this award end up winning a Nobel Prize. So the odds are very good for John, really. And yes, it's really the odds are very good. This is something that is quite extraordinary, and only very few individuals get the privilege to win this award, the Gairdner Award. And one thing about that is that there is a commitment that when you win this award, you have to travel around to showcase what you have done and why you were really chosen for this award. So I'm really pleased to have John today. I'm also using this opportunity to welcome those of individuals online. We are renovating the bigger theater, so this place is not enough to accommodate people, so we're also streaming online. So a brief introduction. It was quite difficult for me to really think about how to introduce John. So I did something. I got all the things, and then you know what I did? I told ChatGPT to tell me how I can introduce John in a very, different way from what people do. And ChatGPT came up with something that was quite interesting. And that is what I'm going to use to introduce John. Dr. John Dick's journey from a small farm near Elm Creek, Manitoba, to one of the world's leading cancer researchers is a testament to his passion for discovery. Initially trained as an X-ray technologist at Red River College, Winnipeg, Dr. Dick's curiosity led him to pursue a PhD in microbiology at the University of Manitoba. I think last year when he was getting inducted in the Canadian Medical Hall of Fame, our vice president was there too, because we are always claiming those who are from us here. So, and I was there as well. Yeah, I was there as well. And then later a postdoctoral fellowship at Princess Margaret Cancer Center. Today, Dr. Dick is a senior scientist at the Princess Margaret Cancer Center and professor of molecular genetics at the University of Toronto, where he has made some of the most significant contributions to modern cancer biology. His development of the widely used xenograph systems and his discovery of leukemia stem cells changed how we understand cancer and initiated the modern era of cancer stem cell research. So initially, people didn't understand why when we treat cancer, there is always a relapse. And it was John's work that actually identified that there are stem cells, and if many of the molecular biologists and cell biologists here understand, stem cells can differentiate and they are quite difficult to kill. And it was John's discovery of the stem cell that led to our knowledge and also led to differences in the way we treat cancer today. Recognized internationally, Dr. Dick has earned over 30 major awards, including elections as a Fellow of the Royal Society of Canada, Royal Society of London, UK, AACR Academy, and an international member of the US National Academy of Medicine. Other awards include the Dameshek and Thomas Prizes from American Society of Hematology, Outstanding Achievement in Blood Cancer Research Award from the American Association of Cancer Research, and the induction into the Canadian Medical Hall of Fame in 2024. His work continues to shape cancer therapy and inspire new generation of scientists. When he was inducted, actually, it was quite amazing. I watched, and they have to do a procession, and they have to wear this gold medal, real gold medal. And I was thinking, okay, maybe when I grow up, I want to wear one of those medals. So, John, the floor is yours. Thank you.
Thank you. It's a real honor and a privilege and a pleasure to be here and to talk in my alma mater. And it was also nice that I could come here a little day earlier and I could meet my siblings and in-laws and other friends who are here. So it's nice that I was able to. It's always a pleasure, to be able to do that. So yeah, you actually, gave a little of my origin story, which is fine. Then I don't have to bother doing it, and I can just get into what I want to talk about. But yeah, so I had a very auspicious beginning that would not have predicted that I would be a biologist going forwards. That little town that I grew up in had a one-room school, and me and Stan were in grade 1, and it was a very interesting origin. And then later on, I ended up doing high school in Elm Creek, which was quite a small collegiate, and it had the basics. So science, chemistry, physics, math, and French and English, and that was also a challenge. I would say I didn't have any biology. I also had the other challenge that my brother, who's up there, was my teacher, and having your brother as a science teacher, not a good thing. So, John, did you do your homework? No. Are you going to do it? Yeah, maybe. Anyway, so. But I took this X-ray course, and I got exposed to anatomy and physiology, and that just lit a bug in me. And I was living with a bunch of guys who all were at U of M, and Animal House, right? 5 guys living in a 2-bedroom apartment. And I thought, well, I'm going to go to university for a year or two, and a year or two led to 10, and the rest, as they say, is history. So I did my postdoc in Toronto with Alan Bernstein, and learned a lot about stem cells, and that was the mecca for stem cell research, as I'll try to explain in a second. And after that, I got a job at the Hospital for Sick Children in the genetics department, and after about 15 years there, I ended up moving to Princess Margaret Cancer Center, where I am today. So what I want to do in my talk, let me see if my pointer comes up. Yeah, there it is. Okay. In the talk is to I'll give you a little bit of background of things that are published. But actually what I'm going to do is I'm going to talk about unpublished work. So for the trainees or people who are interested, you're going to see the latest and greatest that's coming out of our lab. And hopefully it's something that inspires you, and hopefully I can do it in a way that is tractable to a diverse audience. So these are the 3 things that I sort of want to talk about. So I think it's important for us to recognize, I tell the guys in my lab, none of us, research and science is like building a brick wall. And none of us build our brick walls starting in midair. We're always building on a foundation that's been laid before us. And in my case, in terms of stem cell research, it was laid, 30 years before I did this work by Jim Till and Bun McCullough, who were leading scientists at the Ontario Cancer Institute in Toronto. And in the 1950s, they started this work. And so what was going on in the 1950s? Well, the 1950s was, of course, the radiation era, right? The atomic bomb had just been dropped. And people were very concerned about, how to protect humans, but also how to exploit research on radiation. In fact, Canada has a huge tradition in bringing forward, the cobalt bomb was first developed in Saskatchewan and then brought to Toronto and became the leading therapy going forwards. And so there was a whole department of biophysics, which was essentially trying to understand how radiation is impacting on biology to harness it for therapy, but also just to understand it. And so this is the background to which they were working. And so be careful, I could give you a whole lecture just on this slide, but essentially, if the idea that there were stem cells in the blood, so the blood has many parts to it, right? We all know there are red cells, there are megakaryocytes, which make platelets, which allow us to bleed and clot, or control bleeding and clotting. And then there are other components of white cells that help fight infections, amongst other things. And then you have the immune system, T cells and B cells, lymphocytes, which we've all come to love after all surviving COVID and getting responses to COVID vaccinations and things like that. That's the immune side that's doing that. And so at the time of the great experimental pathologists of the 19th century who were staring down a microscope, they all kind of worked out a lot about the features of blood just by looking at what cells look like down a microscope. And they had found cells that were primitive and cells that were more mature, and they kind of worked out trajectories that primitive cells, mature cells came from primitive cells. And if you look in textbooks, in fact, you look at textbooks into the 1950s, it was clear that people were predicting that there were stem cells for each of the different blood lineages. So this is what I want to kind of get across a little bit is how serendipity happens in science, both in this work but also in work that that we've done. Science is much less. I have a vision of where I want to go, and I'm just going to go to that place. It's more meandering and trying to have good eyes to see things that might be important when you look at different things. So what Till and McCullough were doing is they were just figuring out how radiation affected blood cells. And at the time, they wanted to test how sensitive they were to radiation. Now, what people knew before this was that if you take, for example, a mouse and you expose it to radiation, a certain dose of radiation, that mouse is going to die in about 8 days because it's lost its blood system. So the blood system is the most proliferative tissue in your body, and it's the most sensitive to radiation. And so what they were trying to figure out, well, which cells is it and how sensitive are they? And what they did is they took some bone marrow. Oh, and the other thing I should tell you, in the 1940s, they also figured out, I take that mouse that's going to die in 8 days, but if I take a mouse of the same species, so it's not going to be cross-reactive, right? There's no immune system differences. I take another mouse, I can transplant in the bone marrow, and the bone marrow will rescue that mouse. So the blood will regenerate, and that mouse will live fine, and it'll be regenerated. So they had worked out part of the basics. What Till and McCullough did was they irradiated bone marrow at different doses, so from high down to low, and injected it into mice, and they wanted to see what was happening to these blood cells, and they were just going to track them around the mouse body and find the blood cells to see where they were landing and how sensitive they were to radiation. And one of the things they did is they looked on the spleen. Now, a spleen of an irradiated mouse is just a tiny little, 5 millimeter long, very tiny little tissue. And they noticed that in some of the mice, they had these lumps on the spleen. And this is actually an example from one of their pages. And what they realized, and this is where the insightful bit was, that there are differences. Some mice didn't have any, some mice had a lot, and some mice had intermediate levels. And what they realized was that the number of nodules on the spleen was correlated to the radiation dose. So those mice that got kind of, let's say higher medium doses, or medium doses but higher levels had few numbers, and those that had lower doses had higher numbers. So there was a relationship between the number of nodules and the radiation dose. And because of some math and statistics, that told them that that each of these nodules must have been started by a single cell. And when they popped open the single cell, or the single nodule is composed of about 1 million cells, and when they looked on a microscope, it had all the blood lineages in it. And so if you put those two things together, this nodule started from a single cell. That means a single cell is making all of these lineages. And so this was the first demonstration that this model isn't right. There isn't a stem cell for each lineage. There's actually one, and it's able to give rise to everything. And then they went on and did other things. They cut these things apart and did it again into a second mouse and waited for 14 days, which is when they did this. And they saw that some of these colonies were able to redo that, and that was called self-renewal, which is the key property of a stem cell. So many of the cells in the body here are very short-lived. Our red cells are about 120 days. Some of these white cells have a lifespan of 24 hours. So they need to be continuously regenerated. And so the reason the blood system doesn't just exhaust is these stem cells have the ability to self-renew. And that simply means that when this cell divides, even if it's dormant, but when it divides, it will make one daughter cell, which starts a path. Actually, I'll go to the next slide. And so what that led to, as other people started to work, this sort of tree where this multipotent blood stem cell starts to get more and more specialized. The first is into sort of cells of the white cell and erythroid and platelet lineage, and then the lymphocytes down here, and then they get more and more specialized. So that was sort of the idea and theory of what was going on. And so so what that then led to is that this cell, when it divides, one cell actually retains fate of being a stem cell, and the other one goes down the lineage, so that the stem cell is always renewed, and that's the key feature. So they did that work in 1961, and for the next 30 years, almost 30 years, most of the things we know about the blood system was known about the mouse. And what was not clear was whether the human was exactly the same. Now, we knew that there were human stem cells. We knew they could be transplanted. And in fact, there was beautiful work from Don Thomas in Seattle and others who in the 1950s also started to irradiate people who had leukemia and to see whether they could regenerate blood from a donor into those people. And they needed to figure out matching, and you needed cells from identically matched people. And they ended up figuring out how to do that, and so the first person who was transplanted was in 1968, and I think that person is still alive. So now, today, there's about 50,000 people around the world who get this as curative therapy for their leukemia. But the reality is that only 1/3 of people who need a transplant actually have a related donor, because our families are smaller and we don't have enough people who are related to us who we might match to. And so there's a need to understand stem cells to make it more useful for everybody, and there's many things you can do with stem cells. But at the end of the day, a mouse is not a human, and we need to figure out how a human system works. And so when I started my lab, I had done work on the mouse system, and I was staying in Toronto, and I wanted to differentiate myself from my mentor, Ellen Bernstein. And so I thought, well, I'm going to start studying human stem cells. And so the paradox is that I can study these downstream cells here in a lab dish. I won't go into the details, but I can study these in a lab dish. But this was true in 1960. It was true in late 1980s when I started this work, and it's true still today. These are powerful cells. They can regrow the blood in a person, but we still don't know how to harness them and grow them in a lab dish. And that means it's hard to study. And so what I was fortunate in that I had a colleague cells in a mouse, and it turns out there's enough complementarity between, if there's no immune system to resist, there's enough complementarity between what a mouse can provide and a human cell needs, so you now have a mouse running around that has human blood in it. And that, this is a good experiment to do in the first or second year of your lab, because, we're still doing it 40 years later, so it was one good experiment, and it's lasted our career. But it's become the gold standard around the world, because this is the only way you can really study human stem cells. And so this is the armament of everything we did. And so what do you do? Well, we asked how many cells have the ability to make human cells in a mouse. So we did a dose where we transplanted 10 million cells, 50 million cells, 10 million cells, 1 million cells, 100,000. And it turns out you need all the mice at 50 million get engrafted, none of the mice at 100,000, and about 30% of the mice at 1 million. And that told us that only about 1 in a million cells has the ability to make blood in a mouse if you just take bone marrow from somebody. And so, we wanted to work on markers to really gain some understanding of what these cells did. And so it took us 20 years to improve the assay and to carry out these studies, and we and others in the world. And one of the things we knew was, I'm just going to go here. So this is just, the details here are not that important, but it just means that we can take blood cells, the million cells, and we can run them through a sorter. And a sorter is an instrument that allows you to capture cells where you're monitoring them based on proteins that are on their surface. So a red cell has different kinds of proteins on its surface compared to a lymphocyte, compared to a stem cell. And two of the markers that are on the main marker on a stem cell is this marker called CD34. And that marks about 1%. So that right off the bat buys us a great purification, right? So we can take our 1 in a million and we can get it down to 1 in 10,000. But of course, 1 in 10,000 isn't 1 in 1. And so with adding on further markers, what people had shown, we and others had shown, is that these cells had a frequency of about 1 in 20. Group who claimed that work said that was the pure stem cell. We did this experiment. We showed that there are still stem cells down here, meaning that this wasn't the best marker, Thy1. And we came up with another marker called CD49F, which doesn't matter, it's an integrin. And we could take this population and split it into two, a low population and a high, and we whacked them into mice. And what that data showed was that if we take that experiment, take that population and put it into mice, now we get a population of 1 in 3. So we had gone from 1 in a million down to essentially every cell, or almost every cell, in a test tube being the stem cell. So now, we had a test tube in 2011, started our experiments in 1988, so it took a while to get there. And now we could, we pinned another cell onto this diagram. It's like, pin the tail on the donkey, pin the cell in the diagram of blood development. So that was an important advance. And so I'll go back to how we can then harness that and figure out what the genes are that drive those human stem cells. I should make sure I keep track of my time here. So the other part of what we did was that we wanted to study the dark side, right? So I told you about normal blood development. Of course, we get blood diseases, and one of the diseases is, leukemia. The disease that I study is acute myeloid leukemia, which is a nasty disease. If it's diagnosed over the age of 60, which is typically when it is, the 5-year survival rate is 8% or something like that. So it's a really nasty disease. And what that relates to then is, how does this disease start, and what are the features of that disease? And again, this is one of those paradoxes, which is this disease could be running in the bloodstream of a person and going to kill them if they're not treated or if they're not responding to therapy. Yet I can't take that disease and put it in a lab dish and study it, and that creates a complication. How do you figure out what makes it tick? And this is a more broad question about cancer, right? You take a tumor, you whack it out, you mash it up, you look down a microscope. It looks like all the cells are cancerous. And so the question is, if all the cells are cancerous, are they all equal? Well, they're all equally cancerous, but the question is, are they all equally cancerous in their ability, so in their hallmarks, so all the features and properties they're equal in. But the question is, are they equal in their ability to keep that cancer going? So in other words, if I have 100 cancer cells and I test each of them, are they equally each able to keep going? And there was work that had been done years ago where people realized that tumor cells actually, they don't just grow, they die. And so tumor cells are always growing and they're always dying. And so the question is, a cell that's born the day after tomorrow, or if the cell that's born today is dead the day after tomorrow, it's not relevant for that tumor. But if I have a cell today which is going to, keep going 2 or 3 years from now, or I'm going to resist therapy, that's a more important cell to study than the one that's going to die the day after tomorrow. And so that becomes a question, how do you test that? And so we wanted to do this. And so I was starting my lab. We had done the work that I told you on normal cells. And so we wanted to ask, can we put leukemia into mice? Because again, can't grow in a lab dish. And so normal cells do. What about leukemia? Worked beautifully. You can get beautiful human leukemia growing in the mouse. Leukemia can be beautiful, but it's the actual human disease. This isn't a mouse approximation. This is the real human disease. And we did the experiment, 100 million, 50 million, 10 million, 100 million, and to our shock, it was only 1 in a million cells has the ability to regenerate leukemia if you grow it on a mouse. So at one level you can say, well, a mouse isn't a very good thing for leukemia. Like, who cares? Like I said here, maybe it's just a crappy assay. Or maybe it's actually a bit like the normal blood system. And so Sri Lapido, who was a postdoc in my lab at the time, we did this experiment. So here's that famous marker, CD34. And this is what this particular AML looked like. So it had all the expression of these normal markers. So, leukemia is normal cells kind of gone awry, but has some normal features. And so we fractionated all the different quadrants here, down here, 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 and here. And so if we take the red quadrant and transplant it into mice, we take the quadrant down here and put it into mice. We actually take all these guys, I'm not showing that. We put it into mice, and this is the actual real data from the patient, from this paper rather. We put them into mice, and we asked which of these cause leukemia. And what we showed was that if we transplant these, and these, basically 99% of the tumor into the mice, we never got disease. So this is just human, each dot is a mouse, and this is the level of human cell engraftment. You can see that there is no engraftment in these guys in any setting, and that's true for that population, that, and that. But if you take this one here, actually we got leukemia. And so that told us then that this is the fraction of a cell that can actually make that. And when we did the frequency, it's like way more than 1 in a million. It was like most of these cells had the ability to make leukemia. And so this said that there must be some kind of a stem cell for the leukemia, or it's acting like a stem cell, but not making blood, it's making leukemia. And that led to the idea of a cancer stem cell, the idea that there is a stem cell for cancers. Our work done in the 90s precipitated a bunch of work in other solid tumors to see whether it was true. And one of the key pieces of work was this work in 2003 from Peter Dirks, my colleague at Sick Kids who worked in brain cancer. He did exactly the same thing in brain cancer. He knew about our work and he basically tried to repeat it. And he showed that there are some cells in brain cancer which are able to keep brain cancer cells growing. And they have distinct properties. And then that led to work we did in my lab in colon and other people this picture you see here, except for the breast cancer, but all these other ones were done by Canadian scientists. So it was a real tribute to our team, particularly in Ontario, that was able to capture and develop this body of work and said it's not just true for leukemia, it's true for solid tumors. And so what that led to is then the idea that these are cancer cells. They have cancer genes that are making them cancerous and not normal, but the genetics, the same cancer genes are present here, here, and here. But these cells have other properties. We call that epigenetic properties, which are giving them a flavor or a feature which gives them stem cell properties in the same way that cell is genetically equal to that cell, but it's the stem cell features that are driving this hierarchy. So this is all fine, and it's all been done by putting human cells in a mouse, and the question is, how relevant is that? And so if these leukemia stem cells are real, they must be the most important cell in that tumor, and they then should be the ones that are most clinically relevant. And so we spent a lot of time trying to test this and spent a lot of money, and I think Armando was probably in the lab when we actually started to do this work. And so we basically took a whole bunch of patients, of almost 100 patients. We fractionated it into those 4 fractions. Here's fractioned here. We put each of the fractions into mice and asked which ones of these caused leukemia, because it turned out that most of them did it from this fraction, but leukemia is variable, and each of these patients had a different spectrum. Sometimes it was here, sometimes here and here, sometimes here or here. So each patient had to be tested individually, so it took many hundreds of mice. And at the same time, we captured the gene expression programs of each of these sorted populations. And so at the end, we could link together all the gene expression of the cells that didn't make leukemia and the cells that did. And that led to a set of genes that was expressed in a leukemia stem cell specific way. And then we basically said, all right, how can we show relevance? And so we said, well, if a leukemia stem cell is important, we should see it in people, and we should be able to predict something important for that disease. And so we had access to, in this case, data of almost 1,500 people, where leukemia samples were taken, their gene expression was captured, we know what their clinical features were, and we took some complex machine learning algorithms to try to ask how many of these leukemia genes are predictive of something important. And the thing we asked that was important was a question which is sort of unambiguous, and that is, are you dead or alive? And so we asked, can we predict with leukemia stem cell signature whether somebody is dead or alive? And so we trained the signature and then tested it, and this is just the downstream example. And so if you have a high leukemia stem cell signature, you do much more poorly. This is a survival curve, and you do much more poorly than if you have a low signature. So there's something important about leukemia stem cells, not just in our mice, but they're true in real people. And so we've tried to do this across my lab for many years, is to take our basic research and translate it into practical clinical information and clinical data. So to move on, then I'll just give you another version of sort of how serendipity has played a role in the progression of what we've been doing in the lab. So we've been very interested in understanding. A person comes into the clinic, they're diagnosed with cancer, and they get treated, and it looks like the disease goes away. And all of a sudden it comes back and it relapses. And the question is, are there properties of leukemia stem cells that might underlie that? And one of the things we had shown was leukemia stem cells, so many cells in leukemia are growing rapidly. So when you give chemotherapy, they just melt away, and the disease looks like it goes away. But it turns out leukemia stem cells actually can lie dormant. And so they could be swimming in a sea of chemotherapy agents, but because they're not dividing, they can survive. And then because they're regenerative, they're stem cells, they can regrow the disease. And we wanted to generate proof of that. And one thing we wanted to do is to do a genetic analysis of a same patient at diagnosis and a patient at relapse. And We have a very beautiful biobank that my colleague Mark Minden developed in Toronto. Has, I don't know, 50, 60, 70,000 samples in it, vials in it. Patient comes into the clinic, they consent, and a sample is taken and frozen away, and then they get treated, and they either, are successful, or sometimes, years later they relapse, and then the relapse sample gets put into the biobank. And so we went back to Mark, and I should say, I'll come back to this in a second, as to why we were doing that study, because what the results of that's how we started, and that led us into answering a whole different question, which is, person walks into the clinic, the question is, what happened the day before, the month before, the year before, the decade before, right? When did that disease start, and in what cell did it start, and what was the sequence of events that led to the cascades the person walks into the clinic? But we started this by studying diagnosis and relapse. And so we had 12 samples, and they had the biobank, and we had their diagnostic and the relapse sample. Then the picture I need to tell you is when you do genetic analysis, you have to have a germline control. Because if you have a mutation somewhere in your DNA, that mutation could just be you. You were born and you happen to have a mutation. So we need to know it's a mutation in your cancer cell and not in you. And so we just take a control. And typically, if you have a blood cancer, sometimes if you have a colon cancer, they take your blood. But if you have a blood cancer, they just take a little swab of your cheek, the old 23andMe or that kind of thing. Enough DNA in your cheek, the skin cells, and that's the control. But these are patients who would, they've been long dead, actually. And so we just happen to have their samples. So how are we going to do a germline control? And so we thought, we're clever. What Mark banked in his biobank is a leukemia sample. And typically, 80% of the cells are leukemic, 20% of the cells are normal. And in the normal are T cells. Now, T cells happen to be part of the arm of your immune system, which is very different than myeloid leukemia, which is what we study. And we thought we were being clever, and we would just take these T cells as our germline control. And it worked beautifully. I'm not going to show you the data, except that this was at the very beginning of sequencing, and the company we had to make the sequencing panel made a mistake, and we had run all this through. We had the first data, and they said, we have to redo it. And so, we're going to put it was a failed experiment. But Laurent Slush, who was a postdoc at the time, who's an expert geneticist, he said, I'm going to look at the data myself just to see if there's anything there. And it turns out that it worked beautifully for 100 genes. 3 genes didn't work. We had to repeat it. But for these 100 genes, what we would do is we would look at the T cells, and they would be negative for the gene, this leukemia gene, and the leukemia cells would be positive. And it was different for every patient. But he said, I have something weird because there's 3 patients in the set where the T cells seem to have a marker. They seem to have the same gene as the leukemia cells. Now, they're rare. It would be like 1 in 1,000 cells here had the gene. Here, every cell had the gene. But it was identical. And so the question is, how did this happen? How did a normal cell... Initially, we thought we just contaminated our T cell fraction. Our sorter wasn't accurate. But it turns out that we could prove that it wasn't. So the only option we had was to say, well, somehow that T cell must have picked up that gene somewhere back in time, maybe decades before, and there must have been a common ancestor because that led to both of these arms. And so the question is that true? And the question, what would that cell be? Well, if you just go back to the map I showed you, T cells are way over here, myeloid cells are way over here, and the only way they're linked, if you go back up this tree, is up at the stem cell. So the only common ancestor is a stem cell. So we said, okay, well, we have this blood sample, 20% of the cells are normal. Let's dig into that 20% of cells. Can we find stem cells? And it turns out that we can. So we sorted out the stem cells and all these other lineages, and then we did a gene test, and we asked, is the gene present or not? And if it's present, is it present alone, or is it with another cancer gene? And if you look at the leukemia blasts here, they're positive for both the gene we uncovered and this other cancer genes equally, both of them are present. But if you look back at these guys here, they're present, but only one gene is present, not the other one. So, and these cells are behaving normally. They're differentiating. You can see they're making T cells and B cells and myeloid cells. So these are perfectly normal blood cells, but they have a cancer gene in them. And it was just kind of shocking, even more than that, when we calculated the frequency, it was half of the blood was coming from a stem cell that was carrying this gene. And so that meant that what we had uncovered in this failed experiment was essentially a step back in time. It's like, stepping stones across a brook. We were able to look in the normal cells and see that there's a normal cell that actually has the first cancer gene. in it. And what that then led to was two answers to two questions. What was the first cell? It was a stem cell. What was the first gene? It was that DNMT3A gene. And what that seemed to do, that gene, by the way, was causing that stem cell. So normally we think that one blood stem cell won't make any more than 1/100,000th of your blood, because there's about 100,000 stem cells that are active in you and I. But this one stem cell was making half of your blood. So it not only was doing something, it was giving that cell a competitive advantage. It was overcompeting all the other normal cells. And so we were so excited about this because we thought, if we now can look in the normal population, can we find people in the normal population if we just who are on their way to becoming leukemic? If we look at their normal blood, when did that happen? And so we started up this study where we tried to figure out where can we get access to DNA from large numbers of people who are healthy. And so it turned out that in the 1990s there was a lot of foresight, a panel that was started in Europe. And it was a study where there were enrolled Europeans who would fill out a questionnaire, agree to give a blood sample, and they extracted DNA. And they did it on 500,000 Europeans. And they allowed themselves to be tracked over the next decades. And they would fill out questionnaires from time to time. And what they were trying to figure out was, if the industry that I work in, if I work in a chemical industry versus being a bus driver, or if I, if I smoke or I don't smoke, or if I eat, my weight and things. They were trying to figure out what the environmental, nutritional drivers are of cancer. But they had captured this data, and they captured it for a long time. And so we went to them and said, all these people are normal when they enroll. And we asked, have anybody out of these 500,000 people gotten leukemia? And they said, yeah, there were something like 100 people who got leukemia sometime after enrollment. And so we got them, got their DNA samples, and this is the DNA sample when they were enrolled, not when they got leukemia. And then we got 4-to-1 match. We found people who were age matched, sex matched, and everything, who never got leukemia despite follow-up for several decades. And then we did gene sequencing on them and asked, are there any differences between people who are going to go on in the next decade and get leukemia and people who aren't? And one of the things we sequenced leukemia genes, and we showed that there's a way higher incidence of picking up people who have this leukemia gene in their blood compared to people who don't go on and get cancer. But I just want to show you something. These are people who are not going to go on and get cancer, but you can see, 1 in 4 people have this. Have a blood cancer, have a stem cell that actually carries this leukemia gene. And so we looked at this in more detail. And so all the people who are controls, these are people who are not going to go on and get leukemia over the course of 30 years. They typically only have one mutation, and they have it in a particular set of genes. If you are going to go on and get leukemia, when you're still normal, you already have 2, and you have a slightly different spectrum of mutations. You can see some splicing factors here and others. More than that, if you look at the actual mutation, it's much more oncogenic. In other words, you can see it in COSMIC, which is the compendium of cancer mutations. This is like the actual point mutation in the DNA. So much higher in people who are going to go on and get leukemia. And then you can measure the clone size, the VAF, and the expansion is also higher in people who are going to go on and get leukemia. If you put that together, we built a very highly accurate test to predict people who are going to go on and get leukemia. But which is, being sort of beginning to be deployed now, but it's challenging because AML is still a super rare disease, right? 4 per 100,000. Now, if you go back and look at the sequencing data, it turns out that, at least 1 in 10 people over the age of 60, probably 1 in 3 people over the age of 70, have clonal hematopoiesis. So I can go through this room and pick out a few people, like my brother and me, and who are in that, 1 in 3. But it's benign in most people, mostly benign. But there's a subset of people who are going to have those 2 mutations, and they're going to go on and they have an inexorable, rise. And so the question is, for the first time, it's shocking, right, that you could imagine that a disease like AML, which is like a really acute disease, you could think about preventing it. Because here we have a predictive test a decade in advance to be able to do something about it. And I think that this is going to be, important going forwards. Now, and I should say that we did our work at the early part of 2014. Other people had done, in parallel, other work just in other kinds of normal DNA testings, and they, coined this term clonal hematopoiesis in normal individuals. It's not quite so normal. So these people are not going to get leukemia, but it turns out that these people have a much higher incidence of many diseases of aging and inflammation. And one of those is they have a risk. So if you have clone hematopoiesis of a certain type, and we don't actually know it, we haven't made as good a predictor of what it is yet, but in general, the risk, if you have clone hematopoiesis of getting cardiovascular disease, is higher than smoking. So it's a very strong risk, and it has to do, as I'll show you in a minute, with inflammation. Because we have this test, and it's actually been developed into a clinical test at our cancer center, we've been able to run a number of other settings, and we have a very big cardiac center, and so we've been working with Phyllis Bilia, who Lynn probably remembers, and so we've been looking at it in 3 contexts, but we can show that patients who have cardiovascular disease have a much higher risk or if you have clone hematopoiesis, you have a higher risk of getting cardiovascular disease. Okay, still have a bit of time. So now this is the brand new stuff. So that was the old stuff. This is the brand new stuff. So what do we know so far? What we know is that I just told you the risk of clone hematopoiesis goes up with aging. There's something about clone hematopoiesis and inflammation that was a bit obscure, but there's something related. What I haven't told you is that if you have clone hematopoiesis, you have a higher risk of lupus, a higher risk of arthritis, many diseases of inflammation. And as you know, all of those risks go up with aging as well, right? The other thing we know is that this fitness of stem cells goes down. So the quality of our stem cells goes down. I won't go into the details, but just accept that that's the case. And one of the things that happens, I'm going to, in the interest of time, I'm going to skip over the next 3 slides, other than to say that stem cells we stumbled onto this, again, just completely stumbled onto this, that stem cells are more sensitive, even though they're not cycling and more dormant. You would expect that they should resist things, they resist chemotherapy, but they're actually more sensitive to stress. And we stumbled onto this, and this is just an example, we sorted stem cells and progenitor cells and exposed them to a drug which causes proteostatic stress. Stem cells are much more sensitive to proteostatic stress than stem cells are, or than progenitor cells are. And that's just the data there. And it turns out that this is true. This is true for radiation damage as well. Low-dose radiation damage, actually those dormant stem cells are dying more than the progenitor cells. And it's just because stem cells are wired to protect themselves from stress, right? So if you're developing your blood system at the age of 20, let's say a set of stem cells are born, they get activated out of dormancy, they're now active, they could live for the next 50 years, and they could make, trillions and trillions of cells. You make 10 to the 9th you make 10 to the 9th cells every second. It's a massive output that we have in our blood system, almost more than 10 to the 11th cells per day. So you think if a stem cell has damage, it's passing that on to every daughter cell, and they could do it over 50 years. So stem cells have evolutionary logic built into them to prevent them from passing on damage. And one way is that they have high quality control, that if they sense that they're damaged, they'll just kill themselves. They activate the apoptotic pathway. So that is just the premise to say, what's another activation? It turns out inflammation is an activation. When you get COVID or you cut yourself here and you get a big pussy wound, what we often think of is your T cells, your B cells, your myeloid cells are fighting the infection. So we think about the functional ends of the immune system. But actually, it turns out that many cells in the blood system are sensing inflammation. And what one of the data sets has shown is that inflammation will take a dormant stem cell and activate it. And remember what I just finished telling you, stem cells actually don't like to be activated. If they get activated too much, they have the potential for passing on damage. And so that's the basis of what I'm going to tell you next. And so there is nice work in the mouse where if you take stem cells in a mouse and you start to inflame them, they actually start to look like aged stem cells. and that is the self-renewal goes down, and they become more myeloid biased, which we know is a very pro-inflammatory setting. And if it goes on too long, that sets up a cascade to malignancy. But it's kind of a mouse system. It's not clear whether this is true in the human. It's never been studied in this way. And so our entry into this came this way, and that is we took all that stuff I told you at the beginning, our purified stem cells, and now what we want to know is what are the molecular drivers of these stem cells. And there's this amazing advance in technology that's happened in the last just literally 5 or 6 years. And we're doing work today that I never would even imagine just 5 years ago, because we can take individual cells and we can capture their gene expression program. So before we used to have to purify stem cells or other types and then carry out RNA sequencing, which is a way to capture gene expression. But now we can actually take single cells and ask what any individual cell is doing. And this is Andy Zhang, is an PhD student in my lab, brilliant young man, who's carried out this work. And so we've been able to remap human blood development. And so there's labels here, and there's a lot of proof behind the labels. So here's our stem cells, and this is the axis that stem cells take when they differentiate into megakaryocytes or erythroid cells. This is the axis they take when they go into lymphocytes. This is the axis they take into granulocytes and other parts of the monocyte system. So this has become very powerful. We're actually going to be publishing it soon, and it's going to be a really good resource for the community. And for this purpose, we started to study the stem cells. So these cells purified at the very top, 1 in 3 of these cells, very homogeneous, very powerful cells. We take those, did 5,000 of them, individual cells, we map them. Those 500,000 cells all land there. So we have a lot of confidence that this is the stem cell window, and we need to then study the genes of these cells to see what makes them tick, what makes a stem cell a stem cell. And so for the aficionados, this is the approach that we took. So these guys are brilliant. We work in Toronto. You guys remember Jeff Hinton just got the Nobel Prize for AI. So we've been using a lot of machine learning and AI approaches because we have a lot of colleagues that are real expert in this area. And so we can take all this gene sequencing data, it's like, terabytes of data, and we can integrate it and understand it. And I would say, I talked to the high school students this morning. This is like, when you're watching Netflix and you're saying, well, because you liked, this movie, you'll like that movie too. They're recognizing patterns. Or, you listen to this music, well, you'll probably like this music too. So what this is pattern finding, essentially. And so that we're using NMF, which is the program that actually underlies Netflix. And we're driving it with a lot of our own data and all the data sets that we could harvest out of the public domain. Again, for the aficionados, we're taking a lot of sophisticated AI approaches. And I should just point out that Suraj isn't on here, but Suraj and Siam. Suraj started my lab as a high school student, and Siam was a first-year undergrad. And these guys are working with Andy, and they've done a lot of this computational work. And one of the things that came out of this was we took this very pure population of stem cells, and we thought we're going to get one kind of stem cell program. It doesn't matter what these labels are here, but we thought these are the stem cell programs. We thought we'd only get that because they're so pure. But what we got are 18 different features, which was shocking. And the other part that was shocking is you can see there's a label here for the aficionados, right? NF-kappa B, inflammatory response, interferon response, TGF, SMAD. This is smacks of inflammation. And so how is it that these are cord blood cells, right? Completely pristine, taken just out of a cord. Why are we seeing inflammatory programs wired into these cells? And so we wanted to look at this in more detail. And so here's just an example. This is the NMF program. So this is these 5,000 cells, and you can see they're not homogeneous. There's some cells here that are giving the canonical features of quiescence and stemness, but there's cells over here which are wired in with all these inflammatory programs. And it's true with the gene-based, or because we did multiome, which is gene and chromatin, much more powerful, we see it in both of the programs. But the point is it's wired. So then we want to figure out, is this real or not? And so Stephanie is a scientific associate in the lab. She's driving this program, and so Taz is a postdoc, and we collaborated partway through this project with Paresh Vyas's lab at Oxford, and Askar was the postdoc working on that. So these are the guys who did the work. So we wanted to see, is this functional? Right, so this is fine, molecular description, genes are genes, but is it real? So we went back to our system, we took human cells, whacked them in a mouse, let them engraft, and what we did is we tested whether or not these cells responded to inflammation. So we gave them one shot of TNF or one shot of LPS, And we did it at 2 different time points just because we know that these dormant stem cells, when we transplant them, they're all cycling here. And by 20 weeks, they go back into dormancy. We want to see whether a cycling stem cell or a dormant stem cell would respond differently to inflammation. And it turns out that they respond just like a mouse would. This is level of human engraftment. Each cell is a mouse, and here's the control. They have a certain level of human cell engraftment. 16 hours later, there's a marked reduction in the number of human cells in these mice if they get treated with TNF or LPS. So they're responding just as we expect. There's no difference between cycling and dormant. Okay, so now we wanted to ask, okay, so if repeated inflammation in a mouse is giving a damaged blood system in leukemia, how can we repeat that? And so this is our mimic of chronic inflammation. So we let engraftment happen, we hit them at 2 weeks, then we wait, and we hit them again with a second challenge. So just 2 one-shot doses, and then we wait for 20 weeks. So they've had 2 shots and then a long 2.5-month wait. So if I take 1 shot here and wait 20 weeks, they're completely normal. Now, I didn't tell you, but if I just take that 20 weeks and just looked, 16 hours later, it's down. If I look just a week or 2 later, they're back up to normal. 20 weeks, they're still normal. But you can see what happened is if they got 2 shots, there's still a really impaired blood system in these mice. So these cells have some memory of being exposed twice to this inflammatory insult. So we want to understand what the molecular underpinning was. We captured, gene expression, mapped it back, focused on the stem cells. And to our shock, we ended up having not 1 stem cell population, which is what we'd expect. We have 2 stem cell populations. We're not creative. We called one HSC1 and HSC2. And why do we call them stem cells? Well, these are our canonical stem cell signatures, and they only label the tips. So that proves to us that these are molecularly stem cells. And you can see right off the bat, we can actually find that inflammatory signature being wired in, but surprisingly, it's only wired into one of these. The other one isn't sensed by it. And so then we ask, well, okay, if we have 2 stem cell populations and they're exposed to TNF or LPS, who's responding? Do both of them respond or just one of them, or what happens? And so this is the HSC1, and you can see if you respond to TNF, no genes are changing. If you add TNF, these genes are changing a lot. And what are the genes? So this is just the level of intensity here is the response. So you can see right basally, this is already at higher levels. And when you add more TNF, it really pops up. And for the immunologists here, you'll see this is all inflammatory, right? NF-kappa B, JUN-B, REL, MAF, et cetera. Those are canonical inflammatory signatures. So only one population is responding, and it's really the inflammatory stress which is really driving it. And so we've renamed this HSC2 HSCIM for HSC inflammatory memory because it's a stem cell, it's a new stem cell that retains memory of prior inflammatory insults. So again, come to me and say, John, this is all well and fine. This is how artificial is this, right? A human cell in a mouse. Like, what matter does that make? So can we find some human relevance for this? And so one of the pieces of relevance, I'm going to skip over this just for interest of time, just to say the most important the memory response in immunology was really around memory T cells, right? So we all know that we got vaccines when we're like 13 or 14, and we might not see that antigen until we're 50 or 60. It's our memory T cells that are actually giving us a response, later on in time. And it turns out that our signature is highly correlated only with that memory T cell. So there's some convergence there. But what I really wanted to get to is this. Clinical relevance. So is HSC inflammatory memory coming up in any kind of human infection? What did we just finish coming through? Coming through COVID. And so the question is, can we look at stem cells from people who had COVID? That's not easy because people don't want to give bone marrow, but it turns out that there's a group in New York at Cornell, Steve Strzefowicz, who had carried out this beautiful experiment where they had captured stem cells from people who had survived COVID. So these are people so sick that they were in the ICU, and then they recovered. And he got their stem cells for the aficionados, he took 100 mils of peripheral blood and then was able to use 34 selection to capture out the stem cells, so we didn't have to do, bone marrow aspirate. But it was enough to capture stem cells. And then he had people who were in the ICU just for car accidents and other things. And then age-matched controls, because we know inflammation is related to aging. And we ran our signature against it, and it's only expressed highly in patients who survived COVID. So it seems like it has some kind of something real in a real population. We made a signature of his data, his COVID data. It's whatever it is. It's a 20-gene signature, and we mapped it back into our data, and it only labels that HSCIM. So it's very strong proof that this is actually relevant in that setting. What's the other clinical relevance? Well, aging. Now, there isn't a lot of molecular data with aging, but there is some where people have taken, we've contributed some ourselves, taken bone marrow cells from patients who are young, middle-aged, and elderly. And what we've done here is just bin them into young and old. And young is anything less than 60, and old is anything older than 60. Or I guess there's 50 and 60 are the two gaps. And you can see that our IM signatures only enriched in the age HSC. And again, we took all this age data, we made an aging meta-signature of 37 genes, mapped it back onto our data, and it only picks up the HSCIM population. So there's something correlating with aging in our data set. And then finally, remember that clone hemopoiesis thing I told you about, where 1 in 3 of us are going to pick up a mutation in our stem cell. And so the question becomes is, if I have a stem cell and I'm CH, what's it doing to my inflammatory memory? That's one question, right? So is there a correlation between CH? But even more than that, if you think about what I've been telling you, if over time we're experiencing inflammation, more and more of our blood system, our stem cell population is becoming HSCIM, maybe the CH mutation is occurring in an HSCIM population. So how are you going to do that? Because what you need to do is be able to capture stem cells and capture their gene expression and capture whether or not they're CH. So this is like a molecular tour de force. Turns out that my colleague Paresh Vyas has done that with this technology called target sequencing, which gives you site-seq, so you're capturing cell surface and then gene expression. So you can say, here's a cell, it's a stem cell because of its surface markers, and here's this gene expression. And they captured it from people who didn't have CH and people who did. Now, people who did, not every stem cell is CH. Maybe only 20% of them are. And so you can see that there's a stem cell in that person that has the marker and a stem cell that doesn't. And so if you just want to do the basic comparison of the data, we looked at their data, and sure enough, there's a massive enrichment of our inflammatory signature in the patient when they get CH. So there's something about the CH mutation and inflammatory memory. And so we could do this comparison. What I showed you was this comparison. We could also do this one. What about the non-CH-bearing stem cell in that person? How does it compare to somebody who doesn't have it? And it turns out that person is also, that's this one here, that's also highly enriched. So that's kind of weird, right? So this one has the gene, and it's highly correlated to inflammatory memory. This one doesn't, but it's also correlated to inflammatory memory. It's like it by being exposed to it, it's acquiring this phenotype. But the part that became even more surprising was this. So I told you inflammatory memory is highly correlated to TNF signaling. I've showed you a few slides of that. But when we looked at the details of our data, yes, inflammatory memory is highly enriched, but when you do this comparison between that stem cell and that stem cell from the same person, it turns out that inflammatory parts of inflammatory signaling are actually downregulated. When it goes in this direction, it's downregulated. So it's like signaling has been attenuated in the presence of the mutation. And this raises a really interesting hypothesis, and that is that maybe what's happening is that somehow the presence of the mutation is occurring in an HSCIM. And remember, HSCIM, I didn't actually tell you, but HSCIM being driven into dormancy. So the more and more stem cells are exposed, HSCIM are protecting themselves, not by killing themselves, but they're protecting themselves by being put into dormancy. But you can see what's happening is if this happens over a lifetime, if 1 or 2 of my stem cells get put into dormancy because it got exposed to too much inflammation, that's okay. But if 100,000 of my stem cells get put into dormancy, that's not good for me as an organism. And so what it seems to be happening is that when these cells pick up the mutation, it's relieving them. Like if you look at this, cycle prime. So this means that these cells are becoming upregulated. They're becoming more activated. They're being pushed out of dormancy. Interestingly here, these cells are acquiring a young signature. They're losing their old signature. So it's like these cells are being regenerated or rejuvenated by being in the presence of the mutation. So this starts to explain, this picking up the CH mutation over time isn't just a random thing. It's being picked up because inflammatory memory, actually I have this slide here. So what we're saying then is that the first cell here is not just any old stem cell, it's a stem cell that's been created because of a lifetime of exposure to inflammatory stimuli. And inflammation then is the first hit, it's not a gene. These cells are adapting to that, they're putting themselves into dormancy. But then if they acquire a mutation, that mutation relieves them of that negative consequence, and that's what then sets up the cascade either to, in this case, pre-leukemia, but also could set up the cascade for many diseases of human aging, right? So many diseases of human aging are linked to inflammation. And so we think that this actually could be an underlying mechanism that might underlie, the increased fibrosis and, heart disease and other kinds of things that one gets with time. So we're really, excited about this work and trying to figure out, how does this link to patients with arthritis or obesity or other drivers of inflammation? It might be that we're actually reprogramming our stem cells and our whole blood system is being reprogrammed to respond to it. So, bit of hard science at the end, but I just want to acknowledge the people who did the last work that I talked about. But I want to acknowledge more, an award is a weird thing, right? It comes to an individual, I get to stand up and people clap and I wear the gold medal and all of that. But, it's these people who are the ones who did it. We did it together, right? This is what I say at the beginning. Whenever we have a finding, or whether we want to take a direction, or even when we get data, what does it mean? This is Lorna, who's right there rather, and this is our family and grandkids who play a big role in that. So, thanks very much for your attention.
We have time for some questions. I would like to please go to the mic because we have streaming so people can hear your question. We have 2 mics over here. Yeah, unless you can, people won't hear you.
Go ahead.
It really blew my mind, and thank you so much for this. I felt like we've been through a 10 bending, that you downloaded all those knowledge that is accumulated over the half a century into this beautiful crafted presentation. Thank you so much. So I'm a medical geneticist, so my question is very specific. Perhaps I can connect with you afterwards to get the protocol. You done the signature where you use the samples for AML patients. Who are deceased, and then you separate out the T cells as the germline to control for that. Can I ask why T cells, not B cells?
We could have done B cells, but there's a bit. So the thing with B lineage development is that there are sometimes gene programs which are a bit promiscuous. So in the case of AML, sometimes AMLs can actually express some B cell programs.
I see.
And so it's actually, it's a bit more difficult, where T cells are much more distinct. And so that's why we ended up choosing T cells.
Thank you. And I need to get that protocol if I can.
Yeah, no problem.
So I guess I have kind of a related question. It comes back to our conversation this morning where, people have discovered these ABC cells, so age-associated B cells. And so just with your parallel with your AML story, I'm just wondering if there are some similarities in the signatures in these ABC cells as the HSC, and could they be lineage traced back to that's the cell of origin for some of that?
I don't know. I mean, this is all really recent data. Actually, I just realized I have the other slide here. This was a sort of a summary. I didn't want to go into the details because I didn't present some of this data. But if you just, for the aficionados, if you look at the bottom, we actually been able to do some genetic lineage tracing in our single cell data because we can relate the identity of a stem cell and then all the progeny sort of genetically in our data. And what we can show is that if you're HSCIM, you actually pass that HSCIM signature to the downstream progeny. At least we haven't looked at B cells, but we can certainly look on the monocytes and myeloid cells. And so it's a way of explaining how you can actually get an ongoing inflammatory reaction. You know, the cells are very short-lived, right? The professional cells that go to an infection are short-lived. So it's a bit odd. How do you sustain a long-term infection? But it could be that what's happened here is that there are progeny of these cells that are actually doing that. We haven't looked on the B cell side. So I don't know.
Okay, thank you.
Any more questions?
What I can tell you again for the aficionados. So I haven't looked at these aged B cells, but trained immunity, of course, is something which is becoming very well B cell sort of phenotype. We actually don't see any correlation or signatures with trained immunity, trained B cell immunity. We can see the memory T cells, but we can't see the trained B cell immunity. Partly the data is not that good that we can access, but anyway, so.
Yeah, John, fantastic talk. So all inflammations are not created equal, right? There are so many different types of inflammation, and there are so many things that are produced during inflammation. How are you going to be able to tease out what is it that is really driving this phenomenon?
Yeah, we've experimentally only looked at TNF, LPS, and IL-6. There's a collaborator actually at Cornell in the lab that we were collaborating with, who had been looking at myeloproliferative neoplasms, MPN patients, who had been treated with interferon. And she has a whole story on how that interferon exposure is governing sort of the myeloid output of those cells. And she has this sort of progenitor population which is distinct. And when we look at her signature, we can see parts of it embedded in ours. So it looks like interferon is also, it's a different signal, and it's not all of it, but it's a part of it. So it's possible that we're only capturing one element because of the drivers that we're using. That's one. The second thing we'd like to be able to do is ask, can we modulate this, right? So if patients are on TNF blockers or IL-6 inhibitors and so forth, can you reverse this? And so we're just literally just starting. We have a meeting in 2 weeks with the guys in our arthritis center to try to get access to patients who are on some of these anti-inflammatories to see whether we can modulate these signatures. But that's it's just a thought process at this point.
Yeah, hi, Henry Gabalawi. I'm a rheumatologist.
Okay.
And we study the preclinical stages of RA. We have a good handle on that. Now we have biomarkers, autoantibodies that are detectable months to years before the disease starts. And then you get this kind of transition from benign autoimmunity to an inflammatory process. Seems to me that would be a wonderful system to look at individuals who are autoantibody positive, don't have disease, and then they develop ongoing chronic inflammation to see if those adaptive responses
Are you guys biobanking?
We are.
Yeah. All right, we'll have to talk.
So thank you, John.